Hello and welcome to my channel Sam Biology Class. Friends, today we will discuss about site-directed mutagenesis. Before that, will you please consider subscribing my YouTube channel? Your subscription is often a lot to me. Mutation is an alteration in the nucleic acid sequence of the genome of an organism, a virus, or extrachromosomal DNA. It can be spontaneous or it can be induced. Spontaneous mutation results from errors during DNA replication or due to natural chemical reactions in our body such as depurination, deamination, and others. Mutation, on the other hand, can also be induced by certain chemicals, different types of radiations, and also by virus. Mutation involves substitution, insertion, or deletion of base pairs. Mutation can be silent, in which the function of the gene remains same. The function is not altered. But in some cases, due to mutation, the function of a gene is altered. In other cases, the gene is disabled. For example, when there is a premature stop codon, translation results in the formation of a truncated non-functional protein product. Mutagenesis is the process by which mutation occurs. Now the question is, what is site-directed mutagenesis? Site-directed mutagenesis, SDM, is a molecular biology technique that is used to introduce precise targeted mutation at a specific predetermined location of the nucleic acid sequence of the genome. This technique has several applications such as protein engineering and design, genetic engineering and functional analysis, molecular cloning, and also agricultural and commercial applications. As already said, by SDM, we can study the function of a gene. First of all, the gene is inserted into a suitable cloning vector such as plasmid, which includes PBR322, PUC series of vectors, P blue script, and cosmids. In addition, M13 phase vector is also used for site-directed mutagenesis. Other specialized vectors such as P-alter and T7 expression vectors are also used for specific purpose. This figure illustrates the principle of site-directed mutagenesis. So this is the specific gene whose function we want to investigate. So here the target gene has been inserted into a suitable cloning vector which should have an origin of replication, a selectable marker gene, and a multiple cloning site. After insertion of the target gene into the plasmid, the vector is melted or denatured by raising the temperature to 95 degrees centigrade. Such a high temperature will break down the hydrogen bonds between complementary nucleotides that will result in the separation of two single-stranded circular DNA molecules. In site-directed mutagenesis, we are incorporating the targeted mutation into the specific gene by using a pair of oligonucleotide primers which are between 20 and 45 nucleotides long. This primer should be complementary to the DNA sequence around the point of mutation so that it can hybridize with the DNA sequence. In this case, this is the sequence of the primer. This is the 5' end and that is the 3' end. C is complementary to G, G is complementary to C, C is complementary to G, but T is not complementary to G. So this is the single nucleotide substitution that we are using during this process. Remaining nucleotides are complementary. In this case also, G is complementary to C, C is complementary to G, G is complementary to C, but T is not complementary to C. So this is the mutation site where one nucleotide will be substituted by another nucleotide. Remaining 3 nucleotides are complementary. This is the 5' end and this is the 3' end. After primer annealing with the complementary sequence, these primers are extended in 5' to 3' direction by using high fidelity DNA polymerases. So this will result in the formation of newly synthesized complementary DNA strand. Here also by using the same enzyme, extension of the primer takes place in 5' to 3' direction. In both cases, there is a nick, there is a single nick. What is high fidelity DNA polymerases? These are enzymes which have exceptionally low error rates. They also feature 3' to 5' proofreading that enable them to correct mismatched bases that may occur during extension. Examples are fusion, Q5 DNA polymerase, PFU DNA polymerase, and co-DNA polymerase. The primers actually carry the desired mutation. These are oligonucleotides which are customizable and can be designed in distinct pattern in order to incorporate different types of mutation into the target gene or any other DNA sequence. The 3 reaction steps such as melting, primer annealing and extension is repeated for 20 to 40 cycles. This will result in the formation of a large number of newly synthesized DNA strands. In this figure, only 2 such DNA are shown. This entire process is carried out by including all these components and 4 different deoxyribonucleotides in a reaction tube. The reaction tube is then placed in the PCR machine known as thermal cycler. The PCR machine then carry out the entire process. Following amplification, DPNL is added to the PCR reaction mixture and incubated for 1 hour. What is DPNL? It is a type 2 restriction enzyme that digests methylated and hemimethylated DNA. Because the template strands is methylated, therefore this can be digested by using DPNL. On the other hand, the newly synthesized DNA, which are non-methylated, remain protected from the action of this enzyme. In this figure, you can notice that each strand of DNA has a single nick. These nicks can be sealed by the enzyme. DNA ligase. Comparing these two DNA molecules clearly indicates the presence of single nucleotide pair substitution mutation in the newly synthesized DNA. The substitution has been incorporated into the gene of interest by using a pair of oligonucleotide primer in order to understand the function of the target gene. The DPNL digested PCR product, which contain only the newly synthesized DNA molecules, are then used to transform chemically competent E. coli cells. The transformed cells are then plated on a LB agar medium and incubated overnight. This will result in the formation of numerous colonies. Several such colonies are taken out and colony PCR is performed on them in order to confirm the presence of desired mutation. Following this, the positive colonies are sequenced in order to validate the mutation. Friends, this is all about site-directed mutagenesis. For more updates, subscribe my YouTube channel, Sam Biology Class.